immature T cell from the bone marrow arrived to the thymus through the blood vessels. After positive selection and negative selection, the two process and then differentiated and maturity. And peripheral lymphoid organ include lymph node, spleen, and the mucosal associated lymphoid tissues. Those organs or tissues, together with immune cells and immune molecular, make up a complex system for immunity, and the function is to against the antigens. Here we mention the new norm antigen. Well, this class will talk about this subject, antigens. Okay, we see the definition of antigen. So the antigen is a substance that combined specificity with B or T cells, antigen binding receptor can then induce an immune response. So we can see here which part of the cells to recognize the antigen. Antigen binding receptors, right? Maybe the antigen binding receptor of B will called B cell antigen binding receptor. We also call it a BCR. And the antigen binding receptor of T cell, we will name it TCR. The BCR or TCR will mentioned so many times in the future class. Maybe this class will mention it so many times. Do you know what the substance? If you could you list some antigens in our lives? Maybe the fungi, the bacteria, and the virus. Yes, the also COVID-19 also is and another microorganism for our body is the antigen. Besides the microorganisms, some foods such as proteins and the polysaccharide and another, and the drugs, pollen, dust. Those substances will to touch our body by the respiratory tract and digestive tract. And then the antigen will into our body to induce the immune response. Here is the diagram to show the antigen to induce the response. On the left, we can see the antigen recognized by the T cell, and binding cause the after the binding, the antigen caused a cytotoxic T cell, and then the active cytotoxic T cell and produce the memory T cell for the cellular response. On the right part of this diagram, the antigen will binding and trigger the release of antibody by the B cell. At first, the B cell will produce the antibody, and part of the active B cell will produce the memory B cell too. The memory B cell or memory T cell will play a very key roles in the secondary immune response. So we already know the antigen, the function, and we detailing talk about the antigen, what it is. This class will discuss it from the 6th point. The point 1 is antigen and its characteristics, and the second one is specificity and cross reaction of antigen, and the factor of activity affecting immunogenicity and of antigen, and the classification of antigens, and other antigen-related concepts, and the clinical application of antigens. Okay, the point 1, the characteristics of antigens. The antigen molecular generally possesses two natures. That is immunogenicity and immunoreactivity. The first one, the immunogenicity, is the ability of antigen which can stimulate the immune systems of individual to induce a specific immune response. And the immunoreactivity is the ability to acting which can combine with corresponding AB or sensitized T lymphocyte. Pay attention to the AB. It means antibody, which is produced by the active B cell. How to understand these two norms? The immunogenicity is the first step, and the immunoreactivity is the second step. Okay, we'll detailing see some example. The immunogenicity is the ability to induce a hormonal and cell-mediated immune response, and induce the production of antibody and affect lymphocytes. We can see we should be understand here. If the antigen could reduce the response, this step is the first step. We can imagine that if this step is loose, the B cell or T cell cannot active, right? So the here the antigen active the B cell or T cell is the first step to response. And resultantly, after the active, the B cell will produce the antibody, and the T cell will become to differentiated into the active CTL. This production is responsible for the antigen. It's also called the antigenicity. Oh, immunogenicity, antigenicity. Yes, right. The antigenicity is the ability to combine. Pay attention to here. Combine specificity with the final product of the above response. So we can imagine that what is the function or what should be active the T cell or the antibody produced for what? For combined specificity with the antigen, right? So we can see another diagram. The AG it means an antigen. It with a special structure here, a triangle. The black triangle named means a special structure here. And the AG, the antigen, is for the the antigen will inject into the mouse, and the mouse will the B cell or T cell in the mouse will be active. So this process named immunogenicity. And then the T cell or the antibody will combine with the AG, also called antigen. The T cell and the antibody combined with the AG specificity is the named antigenicity. If the immunogenicity not exist, the antigenicity also not happen, right? So these two processes is the first and the later two steps. So the immunogenicity and the antigenicity are the two processes of the same event. Is a closed loop. Closed loop. And the second part, I think, specificity and a cross-reaction of antigen. Here we talk about the specificity. What means the specificity in the immune response? We can see here there are two keys named A and B, and a receptor. Obviously, the A the key A convenience for the antigenic determinants. What is the epitopes? The epitope is the part of portion of antigen molecule which can be specifically bound by the antibody and antigenic receptor of left side. Maybe it is 2 or 23 amino acid residues, and 6 to 8 nucleotides. Different structure or different components displayed different structure of the epitopes. In the biochemistry, we should know the structure of the amino acids. At the first of at the first step of the production of a protein, it is a primary structure. The amino acids one by one combine and bind each other like a line, and then it become to a alpha helix to form a secondary structure, and furtherly is followed furtherly and to become a tertiary structure. If it has one more substructure, they will together for the for the other, I think is fourth, to combine each other and to form the quaternary structure. What is the picture want to show? I think is that different protein will have a special structure, and on the surface of the molecular will to display a special structure, and just the special structure to induce the different antibody. How does it work? We can see here is two antigen, different antigen. The green one, antigen 1, here is antigen 1. Sorry, I mark it clearly. Okay. Here showed two antigens, antigen 1 and antigen 2. And we can imagine that the antigen 1 is a protein 1 to fold, and furtherly to form, and the antigen 2 induce the antibody is responsible for the square epitope. And instruct if the green antigen to bind the antibody which produced by the epitope 2, it can't bind it, and another is the same. So here we showed the sentence under the picture. Antigen determinator is called epitope. Is the site of antibody binding with the antigen binding with the antibody, and a septal change can affect the specificity of antigen. So this is displayed the specificity of the antigen's epitope with the antibody. And another one is antigenic valence. We can see the picture below the sentence. Here we can see an antigen with showed 3 kinds of different epitopes so on the surface of the antigen. And the purple corner means 3 kinds of antibody. Each one kind of antibody responds for the relative epitopes. So we can see the antigen. It also means that the antigen can bind with 3 or more different antibodies due to it has 3 kinds of epitopes. This means antigenic valence is a polyvalent antigen. Polyvalence, the poly, it means more than one. So the most natural antigen are the polyvalent antigen. We understand it is okay. And the classification of the antibody as antigenic determinants. The epitopes can divide it into two I think it's 2 parts. The part 1 is a conformational determinant, and the other one is a sequential and the linear determinants. We can see here is showed the diagram. It showed a protein or amino acid to fold a structure, and we can see here the black is a turn showed the is showed, and two these turns close and it showed as special structure. Here is named conformational determinants. And another part of this structure, we can see the red showed the sequential determinants. It is a single part of a layer, also a determinants. Why it will divided to the 2 groups like this? Because the conformational determinants will recognized with will be recognized by the B cell, and the layer or the sequential determinants will recognized by the T cell. T cell, the red layer, the red determinants will recognized by the T cell. Thymus lymphocytes. And the conformational determinant will be recognized as the A, B, C, D, the B cell. This point we will detailedly talk about it in later. Here is some I think is a description of the conformational determinants. We just know it okay. And the sequential determinants. Here we can see the different kind of epitopes. The right is the left is the layer epitopes, and the right is the conformational epitopes. These 2 kinds of epitopes is recognized by the 2 cell or the 2 combiner. I think it is not difficult to understand this part. Maybe we will talk on the other hand to talk about it. This problem, this question. So here We are according to the type of cell recognized antigenic determinants, the B cell determinants and the T cell determinants. Before we to talk about these two kinds of determinants, we should be understand something about the process of antigen after into the body. If some okay is we, for example, for the COVID-19, okay, if someone unluckily infected by COVID, and this COVID is an antigen into the body, and first the COVID will eat by the APC cell. The APC eat the COVID and then digest it. After the digestion, the APC will present some layer epitope of the COVID to the T cell. Some classmates will be asked, the B cell can't recognize the COVID directly? Yes, the B cell cannot recognize the COVID directly. If T cell to get the antigen, it must after the process of antigen presentation by the APC. The APC cell including three kinds of cells. The COVID will take up by the APC and after the digestion and put the antigen to the T cell. Obviously, after the digestion, the B cell get the information of the COVID is not I think is not a total of COVID. It is just a piece of protein, like a layer. So now you should be understand the B cell. Why the B cell to? Okay, we can see this picture will be understand easily. The T cell and epitope and the B cell epitope. You can see the T cell epitope is the piece of it should be digested, and then one piece a piece of the proteins. But the B cell epitope is a conformational structure. So the B cell can recognize the antigen directly, but the T cell cannot, because the T cell only recognizes the piece of antigen. It should lead the process of digestion by the APC cell, APC. Understand it? Nobody answer me? This part, did you understand it?
Yes, understand.
Okay. We have an example here. The immune response, the epitope that the TCR and the BCR recognize is different, and are called by T cell, but those are called T cell epitope and the B cell epitope separately. Here we can see the layer proteins. It means pancreatic glucagon was was inoculated into the mouse, and reasonably the mouse will produce the antibody and the active T left side, and then this protein in the conformational structure will recognized by the antibody, and the layer part will combine with the active T cell. So this protein will be recognized by both two ways. So here is some transcription about this process. This picture, this PPT I will send it to you, and we understand one protein will recognized by antibody or the active T cell, each other to at the same time, because the protein have different the structure. This structure named different have a different characteristic. The conformational structure named conformational epitopes, and the layer structure named is what is I forgot this name, is a layer epitope. Yes, is a layer epitope. And because the T cell cannot directly recognize the antigen, so it should need the help with the APC, the antigen presenting cell, the help. After the antigen, after the antigen will digested by the APC, and the APC present the piece of the antigen to the T cell. The T cell will be activated. Remember this different. So the T cell and the B cell have a different pathway for recognize the antigens. And here is a detailing picture showed the B cell epitopes. The B cell epitope is recognized by the antibody CDR3. It also means BCR. The B cell receptor on the surface, and the B cell epitope is on the surface of the molecular. It also named conformational determinants. And the T cell epitope is that represented by MHC molecule and recognized by the TCR. The MHC molecule is on the surface of the APC. the MHC molecular is knocked on the surface of the APC. The APC is an antigen presented cell. It includes dendritic cell and the macrophage cell. Here is showed clearly about the T cell to recognize the antigen. Actually, here actually it should be changed the code because the T cell recognizes the antigen is not the original antigen, but an antigenic peptide, right? Because it's a piece of is a small part of the original peptide protein. It is just a peptide. So here we should maybe it not reasonable to call it antibody. We should be call it antigenic peptide. This peptide is present by the MHC2 molecule. The MHC molecule is located on the APC. See, I addressed here. The T cell cannot recognize the antibody antigen. If the T cell wants to recognize the antigen, it must need the help with the APC, because the APC got the antigen and digest the antigen to piece and piece, and then this piece is named here is antigenic peptide. The antigenic peptide and bind with the MHC2 together to give to the TCR to recognize. So I think I was clearly to claim this part. I hope you is already understand it. And the other concept of the sorry， 哎，李老师，哎，哎，啊，您只有这一个问题吗？现在？哦，好，那因为我现在在上课，我等一下上完课给你们把这个东西发过去。哎，好嘞，好，哎，好，开户行是吧？只有开户行这一个问题。好。Okay, here we talk about the other point is a common antigen and a cross reaction. What means the common antigen? And before we talk about the epitope of the antigen. Here we can see the common antigen is the same or similar antigenic determinants that exist between two different antigens is called combination. Okay. On the other words, if two antigen maybe is different each other, and if they have showed the similar or the same epitope, only one is okay. The antigen 1 have a triangle epitope, and the antigen 2 have a triangle epitope 2. The antigen 1 and the antigen 2 like could be called common antigen, just like the picture showed here. Maybe the antigen 1 and the antigen 2 have another kind of epitope. It doesn't matter. Maybe because they already share similar or same epitope is okay. They are called common antigen. So if two different antigens share or have the similar epitope, the same or the similar epitope induce the antibody will combine each other. So just the described here is the reaction that antibody react with the same or similar antigenic determinant between the different antigen is called cross reaction. We can see the picture, the yellow cycle. I think is how to describe the color. Maybe the cycle named antibody, anti-A body, right? Do you see the anti-AB is showed the same antibody? We can see here AGA and AGB and AGC is different, the three kind of antigen. The AGA and AGB share the same epitope, and the AGA and AGC have a similar epitope. And then the antibody A is induced by AGA, but the ATA not only bonding to the AGA, this epitope, it also can band with the AGB and AGC. This process is called cross reaction. Here is the other diagram to description here. The AG1 have three different color of epitopes. After injection, the rabbit will produce the three kinds of antibody. And different another same corner together, the red antibody to bind with the red epitope, and the green antibody to bind with the green epitope. And we can see the below the antigen 2 also have different color of epitope. Maybe here is three kinds of epitope 2. Also, the rabbit will produce three kinds of antibody after inject H2. We can see the AG1 and AG2 shared the same corner, cycle or the same corner epitope, the red. So the red antibody also can bind with the Ag2. Also, even the red antibody is produced by the Ag1. So it is the cross reaction. The antibody is induced by Ag1 and combined with Ag2. Cross reaction. What is the significance of common antigen and cross reaction? We should be know something about this. And the first class we talk about the smallpox. The smallpox is very severe disease for the human. And the general, the England doctor use the complex vaccine to produce to protect the people and divided and avoid to suffering from smallpox. What the mechanism is? Here we should be know it because the cowpox vaccine injected to the body. We can see the cowpox vaccine. There are different two kinds of epitope on the surface of the cowpox. So the little boy will produce at least two kinds of B cell or antibody. This is a very reasonable pathway here, because the compounds have two different epitopes, and the smallpox virus also have different two different epitopes. But we can see the smallpox and the cowpox share the same epitope. The yellow 10 cross shares epitope. So the cowpox vaccine induce the produce to produce to protect our body and avoid from the infection of smallpox virus. Okay, this class we showed the something about the antigens, the definition of the antigen and the T cell epitope and the B cell epitope, and the common antigen and the cross reaction. The time is here is okay. The rest we will be talk about in at tomorrow class. Okay, any questions?
OK， 老师，
Okay.
没有问题，老师。
Okay, thank you. Okay, now this class is over. Goodbye.
OK，
老
老师
师，Thank you.
OK，老师，
Thank you.
Okay.
拜拜。
See you.